And this is my proficiency for DNA extraction. As you can see, I have my unknown bacteria with 2 tubes, and I have my known bacteria that I will be using in this lab. Here is one of the 3 buffers that we will be adding into the tube, which is ATL buffer. You will use about 50 microliters of it. Then here is the second buffer that we will be adding, which is AL. You will again be using 5 microliters. And now you will be using the SDS 1% buffer, and this will be one of the last buffers to be adding. Once we're done with that, you can see that I am performing the wet extraction. So again, I turn on the torch, wait for it to turn bright red. That means that it is sanitized. And then I will open up my petri dish and my tube so I can pour it in there. I scrub some of the DNA on the loop, as you can see. Then I pour it in there to mix it a bit with the buffers. Now in the meantime, I will be discussing about how DNA contains all genetic materials of the organism, and when we are extracting the DNA, we are separating the genetic material from the proteins, RNA, organelles, anything close to the material, and then we will purify it to investigate the genetic material that it contains, and we will investigate the sequences as well. You will then add 10 microliters of protein K and then vortex it and then you will incubate it for at least 30 minutes. You can see here that I'm vortexing it. Once you are done, you will add it into the microcentrifuge for 5 minutes at 2000 RPM, which stands for revolutions per minute. Then when you are done, you will move all of the extra supernatant with a pipette, and then you add ammonium acetate and ethanol, and then you will freeze it for 15 minutes. So then once you're done with centrifuging and then you wait the 15 minutes, you will see the pellets at the end. You will then flip it upside down and then add 50 microliters of ketogenic buffer to the DNA to preserve it, and then you will vortex it again. You will be using these results for your nanodrop that to measure the nanodrop that you will do next week, and then you will use the electrophorus, as you can see later on, and you will set the machine at 130 volts for 30 minutes. Then you will fill up with a TAE buffer, and then you will place the gels that you and your partner made, and then you will insert the DNA with the stains. So you can see here the DNA stains are in the wells, and this is me performing it, inserting the DNA stains into the wells, or into the gel wells. And then the next image will be this gel stains that my partner and I, and it is labeled. Then after performing the gel electrophoresis, you can see that there are some DNA strands, and then here's the successful DNA ladder.